Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing who will be part of the CannMed 2020 conference in Pasadena, California this September 20th through 22nd. I am your host, Ben Amaralt. I'm the marketing manager of Medicinal Genomics and a proud member of the CannMed 2020 team. Head over to CannMedEvents.com to check out our Phase 1 schedule, which includes presentation topics from 20 of our invited speakers and abstract presenters. We will continue to add to this lineup throughout the summer, so be sure to sign up for email alerts so you don't miss any announcements. If you sign up on our podcast page, CannMedEvents.com/Coffee Talk, you will also be entered into a drawing to win 2 tickets to our CannMed 2020 VIP dinner. On this episode, we talk with Brianna Cassidy, who is the Chief Scientific Officer at CDX Analytics. Brianna and the team at CDX perform safety testing on all types of cannabis samples, including potency, pesticides, heavy metals, residual solvents, and microbial contamination. As you will hear from our conversation, Brianna takes a creative and analytical approach to solving problems that come with testing cannabis products. At CannMed 2020, Brianna will share her process for evaluating new analytical methods and technologies for her lab. As you can imagine, there are a number of vendors looking to install their systems in cannabis testing labs, so it helps to have a thorough and objective process to choose the best option. Our team knows firsthand that Brianna is thorough when evaluating new technologies. She and the team at CDX provided data that was a major part of the Medicinal Genomics manufacturer validation document. Brianna and I also discussed the issue of consistency when it comes to potency testing. I was surprised to learn that cannabis flowers from the same batch are often 50% more or less potent than what's reported on the package. She also explained how water content can make a big difference in potency results. We are excited to have Brianna as part of CannMed 2020, and we are excited to see you there as well. Go to CannMedEvents.com now to see our ticket pricing options and get your ticket today. We are currently offering early bird pricing through August 1st. We are also offering a 100% refund to ticket holders should CannMed 2020 need to be postponed due to COVID-19. Rest assured that we are working closely with the Pasadena Convention Center to make sure that we can provide a safe environment for our staff and attendees. And as of this recording, the event is still on. But just in case, we have updated our refund policy, so please check out our website for details. I also wanted to thank this episode's sponsor, The Cannabis Scientist. The Cannabis Scientist is a publication with an important mission: to promote good science in our growing field. Register for free on their website, thecannabisscientist.com, and get unlimited access to expert commentary on testing, extraction, medical research, and regulation. And finally, we want to thank our friends at the Hemp and Coffee Exchange for fueling the CannMed Coffee Talk podcast. The Hemp and Coffee Exchange is a grassroots Tampa-based coffee roasting company creating progressively sustainable consumables combining 2 of the most genetically diverse plants on the planet, hemp and coffee. Check out hempcoffeeexchange.com and use the promo code DRINKHEMP, all one word, to get 10% off your purchase. That promo code, again, is DRINKHEMP. Okay, without any further ado, please enjoy my conversation with Brianna Cassidy of CDX Analytics. Hey, Brianna, how are we doing today?
Doing pretty well, Ben. Yourself?
I'm doing well, doing well. Thank you so much for joining us on the podcast. And as I always like to say when I talk to people from the lab testing profession, I love getting a chance to talk to you because we at Medicinal Genomics, we work with you guys very closely. And I know what you do on the microbial side, obviously, because of what we do at Medicinal Genomics, but I really like to talk to you about all the other things that you test for at the testing lab. So tell me a little bit about CDX, your position there, and what a typical day is like.
Sure. Well, first off, I'd like to say I'm honored to be a part of the CannMed conference. I'm very excited to be presenting there, so I'm looking forward to that. And my title is Chief Science Officer at CDX Analytics, and we test for a myriad of different things in cannabis and cannabis-infused products. Of course, the cannabinoids, the active ingredients, terpenes, also possibly active ingredients, and then some contaminants. So we're looking at heavy metals, pesticides, microbials, mycotoxins, and then we have some disruptive technology in the lab too in regard to our dry weight measurement and accounting for water content in samples.
Oh, tell me a bit more about that.
I had a feeling you might ask. So
You have good instincts.
it's important to talk about water content because the reported concentrations are supposed to be indicative of how much what it is we're seeing is in the dry weight of the sample being tested. And cannabis can be around 80% water. So if you don't accurately account for that 80% and subtract it out, the result from your very precise analytical instrumentation could be completely moot. The suggested way to do this by the American Herbal Pharmacopoeia is by loss on drying. And that's essentially taking the sample and putting it in an oven for a certain amount of time. So that has some implications, that loss on drying method. Number one, water desorption is a very slow process. And as you reach completion, it becomes slower and slower. So if you're to put a really wet sample in alongside a very dry sample after the same exposure to temperature and time, those two samples will still have different water contents. So the more wet sample will still be wet. And if you don't account for all the water in a sample, you are going to artificially deflate all of your analytical results. If you overaccount for water content by possibly, let's say, baking off terpenes and other volatile parts of your sample, assuming they're water, but they're not, then you can artificially inflate your analytical results. So what we've done is we've incorporated a technique other than loss on drying. We're using gas phase Karl-Fischer titration. And what this does is using a subsample of what we're testing, we can figure out how much reaction we get with the water content in that sample using a chemical test, and simply subtract the water content out using the molecular weight at the end. So this allows for an extremely precise and accurate result for all of our testing that we do on flower.
Okay, so if I followed that, and I'm not sure that I did, but that's okay. So what you're doing is actually just measuring the water content and then kind of subtracting that out or applying some kind of algorithm to separate that out rather than trying to bake it to be 80% or whatever percent is the recommended dry weight. Do I have
Yeah,
that right?
exactly. So instead of baking out the water content, we test for it chemically and then subtract it from the result.
Excellent, excellent. Now, when you get a consumer going into the dispensary and buying some flower, what is sort of the average water content there? Does it vary between the different buds or the different strains, or is it relatively consistent?
We see cured bud coming in at around 4 to 10% water content by weight.
Okay, so that's a decent range.
Yeah,
And
and, when you're talking about the testing limits in Massachusetts, where we're quantifying down to parts per billion levels, that range has a huge impact on the final result.
now, is that specifically for potency, or can that apply to heavy metals and other contaminants as well?
Yeah, it's a good question. Every single result that is reported for a flower or plant-based sample is impacted by water content.
Okay. Okay. So this all really seems to jive with the article that I was reading that you and Cindy Orser had authored. That was all about cannabis uncertainty, and how, if you're taking a big batch of cannabis and selecting different subsamples of that to do your testing, the difference in those different subsamples can be pretty vast. And I imagine that this water content's just another variable that goes into that. Fair to say?
Yeah. So that article was a blast to write with Cindy. I think it's, Medicinal Genomics is great about this too, is the collaboration to talk about these variables like, measurement uncertainty, water content. Why is it that people are getting different results for what they think is the same batch? So those discussions are really important to have. And yeah, you're right. That article does encompass a lot of different discussions on what can impact your test results.
And so, in addition to water content, what are some of the other things that can be leading to this uncertainty problem? Is it something that's just sort of a necessary evil of testing something like cannabis flower, or is it something that there's not enough standards between the labs, combination of the two, or more?
Yeah, I mean, you covered a good bit of them, but it is true to say that any measurement that is taken, whether you're measuring a string with a ruler or you're measuring the nanograms of pesticide in 1 gram of flour, both of those situations are going to have a plus or minus around the result. So the article that Cindy and I wrote goes into some other variables that could be adding to this plus or minus. We already talked about water content, and you briefly brought up testing different buds from the same plant and how the plant itself being inconsistent throughout in terms of cannabinoid concentration and possibly contaminants does contribute to the plus or minus of your result. And the most efficient way to mitigate that is to have homogeneous batches of product, meaning that they're well mixed and that they are the same throughout. So that when a subsample of that batch is taken to a laboratory, the results for that subsample actually represent the batch itself.
Now, that's got to be pretty hard to do with cannabis flower, though, because you don't want to, take a huge batch and grind it all up, because then you're gonna, lose your bag appeal, lose those nice beautiful buds that people are so interested in. So how do you overcome that?
It's difficult. And, instead of overcoming it in an industry where that isn't something that is altogether possible, you instead have to accurately account for it. And what I mean by that is, if one bud has a different result than another bud, then understanding that when you look at the label of your product, of your cannabis buds, when you pull a bud from that container to consume, that bud could be plus or minus 50% or more of what is on the label. And it's, that can be alarming, but at the same time, knowing that and understanding it is just as important as solving it.
Wow, 50%. That's that's
Yeah,
significant.
at
And
least.
especially,
I didn't mean to cut you off, but there is a peer-reviewed publication in Forensic Science International where they show the test results of different plants that are within the same clone and how much the cannabinoid results vary from bud to bud. So that's an interesting one to look at if you want to get into it even further.
yeah, and I'm just thinking with CannMed being so focused on using cannabis as a medicine, that's got to be something that if you are using flower to treat your condition, that's something you really need to take seriously.
Yes.
So, what are your thoughts? I mean, I know you're not a physician, and you're not going to be giving out any medical advice, but in terms of patients who are treating with cannabis flower, what are sort of some things that they need to keep in mind?
It's first of all, I appreciate you recognizing that I'm not a medical doctor. Doctor of philosophy is a completely different deal. I would look more towards the products that are available that are a little bit more homogeneous, meaning, liquids, and they all have their own inhomogeneity variables related to them. And until someone does a test comparing the homogeneity of cannabis infused products to concentrates or extracts to flowers, it's kind of hard to navigate that question. But theoretically, I would assume that liquid or cannabis infused product based options are more homogeneous than buds themselves.
Right, but then comes the whole problem of, consistency from batch to batch or lot to lot, which I can imagine um, can vary as well, just because of whether it be sourcing the material or changing in production or things like that.
Yeah, that's true. And in with pharmaceuticals, that's where I come from, is the pharmaceutical industry. The USP has very strict testing requirements that have to be met in order for a sample to be even considered representative of the batch. And those tests ensue taking, 30 plus units and testing them individually, and they have to be within the same result in order to actually move forward with that batch. So I do see the cannabis industry eventually getting to the point where they require homogeneity testing. I have seen clients that are starting to undertake homogeneity testing as just a precaution for their business, because what that does is it gives them a snapshot of how consistent their product is. And it ensures their clients, the consumers of this product, that they can expect that the label represents what they are consuming and that it's consistent as well. So it's actually a big advantage to the marketing of a product to go ahead and undertake this homogeneity testing and to shoot to create a more consistent product.
Yeah, and I imagine that's where we hope this is all going, is that the companies and the manufacturers will do all this testing not because they have to, but because they want to, or because the customer demands it or expects it.
Right.
So it's good to see that at least some are already taking that step, and eventually everyone will get there. So
Exactly.
you mentioned you came from pharmaceutical testing, and I'm curious, when you say that, in the cannabis space, that the difference in concentrations can be up to 50%. Where is it that the pharmaceuticals, what percentage or what difference are they really aiming for in terms of consistency?
So with cannabis, I think the 50% is going to be a relatively normal happening. I think that the concentration can be a plus or minus more than 50%, just to clarify. In pharmaceuticals, they measure consistency by a metric called relative standard deviation. And so there are USP guidelines on, it's called content uniformity, is the title of the document, where they give different um, different relative percent differences based on the product type and how potent it is.
Okay, that makes sense. I did want to go back to, you mentioned when we were kind of talking about alternatives to using maybe cannabis flower for treating a condition,
Mhm.
that there are different cannabis infused products out there. And I'm curious because you see all different types of cannabis infused products out there. I'm wondering what are the sort of the normal ones that come through your lab, or maybe some of the not so normal ones that may have come through your lab.
We see a myriad of different matrices, and we're not generally surprised anymore. We've seen anything from infused drinks. That market is growing, and we see a lot of infused chocolate products, a lot of infused gummy products, and more recently, vaginal and anal suppositories. I mean, the list of products that we see in the lab just is unending.
Yeah, no, it seems like every week, every month they're coming up with a new thing to infuse cannabis into.
Yeah.
What is the most challenging of the matrices that you've had to deal with?
I would say the infused drinks. Um, infused products,
Interesting.
yeah, infused products in general require more astute testing because they include ingredients that could possibly interfere with the test. They could interfere with the test by covering up the signal of the analyte that we're looking for, where we can't see the cannabinoid or we can't see the metal. They could interfere with the test in a way that those ingredients hold on to the constituent that we're looking for and keep it from being in the solution that we're looking to test. And to mitigate that, CDX has launched a formulation test. And what that is we actually go in and we work through the sample in several different methods to ensure that those things, that those possibilities are mitigated. So, cannabis infused products in general, where we're talking about literally an infinite list of possible interferences, have to be taken seriously in testing. And, in any instance where anybody says that they have a one method solves all process, they just simply haven't looked into it in depth enough to find that there are variables in these different cannabis infused products that do impact their tests.
So you basically put any new cannabis type product through sort of a battery of tests to sort of really break down what's in there and what could potentially be throwing off your results.
Right. This is something that we have been working with a few of our clients in doing, and this is actually part of what I plan on presenting at CannMed. So it's going to be a demonstration of how different methods can give different results on the same exact products, and understanding how to take that knowledge and mitigate in order to report an accurate and precise result.
What an excellent segue.
Yeah.
So, the title of your talk here is Navigating Cannabis Method Options Through Implementation of Novel Validation Design and Statistical Comparison. And you did just give a little bit of a preview with about what that talk is going to be about, but I was wondering if you could give us a little bit more.
Absolutely. So we have a lot of different testing options as third party testers in the cannabis industry. A lot of instrument vendors have prepackaged methods and instrumentation that can be used to measure the cannabinoid concentration or the heavy metal concentration, or, as you very well know, the microbial concentrations. And the talk is going to be about how to choose the method that is most appropriate for what it is that you're doing in a quantitative and statistical way, because accurate science is all about absolving subjectivity as much as possible and looking at the data and the relative standard deviations and the robustness. That's what's going to give you the answer to which platform you should choose to do your testing.
Excellent, excellent. So what are some of the different methods, or I shouldn't say methods, but what are some of the different types of testing that you are going to be sort of highlighting in your talk? Is it more on um, the potency side, heavy metals, microbials, a little bit of everything?
We find different avenues every day that we're in the lab. So what I plan on doing is choosing the ones that best illustrate the need for quantitative decision making. And right now, it's looking like that discussion will be based on microbial testing. There was a little bit of a preview to this given by Yvonne. She's already presented this last fall with the Grim Reefer, and so I might build on that a little bit with the in lab testing we did at CDX and the validation that we have for inter laboratories. But I have a lot of different options in terms of what other test methods I include. Could be a little bit on the water content quantification we talked about earlier, comparing that to the loss on drying method in terms of precision and accuracy and robustness. I could talk about cannabis infused products and different extraction methods for cannabinoid testing and how those impact the results. It just goes on and on. It's such an exciting new industry. Scientists have only had 20, 25 years to work with this matrix. And it's really fun to be a part of, battening down the hatches and coming up with methods that consistently trump what was currently in use days, weeks, months, or years before.
We're going to have to block off a whole afternoon for you.
I don't know about that, Ben. You'll have to talk to AJ.
No, but it's I'm glad that you brought up Grim Reefer and the talk that Yvonne had given at CannMed 2019, because you did contribute a lot of great data that was used in that, and we want to thank you very much for that. And if folks are interested, they can check out that talk and all the CannMed 2019 talks at canmedevents.com. Nice little plug in there for that.
Yeah.
So you mentioned what you did before you got into the cannabis space a little bit briefly, but I was wondering a bit more. What did you do before you got into cannabis, and why did you decide to make the switch?
Sure. It's been a long journey, but just very quickly, I have a chemistry bachelor's with 2 art minors, art history and studio art. Um, After that, I got my PhD in analytical chemistry. And during that time, I worked on a lot of forensic applications, blood detection, blood stain age development, using different light based techniques combined with statistical data analysis to correctly classify unknowns. A lot of really fun stuff. I also did environmental sampling and testing. And from there, I moved on to the pharmaceutical industry, where I worked to identify the impacts different formulations have on active pharmaceutical ingredients. So essentially, what I did was keep track of what fillers in drugs do to the drugs themselves and how they impact the expiration date of the drug. So that work, yeah, it turned out to be incredibly relevant, especially to cannabis infused products. And I'm really excited to apply that. But in getting into the cannabis industry, it was something, like I said, it's so new. People really haven't had time to nail down the testing methods, and there weren't that many peer-reviewed publications, and still aren't, on cannabis testing. So that was very lucrative to me. I thought it would be neat to be a part of something where creativity paired with chemical knowledge could be used to really give back and have a high impact to society. So that's what I did. I worked at CDX Analytics for nearly a year with state-of-the-art brand new instrumentation, designing methods. And I'm very grateful that the president and CEO of CDX has a PhD himself, because I don't know any other businessman that would give me that long to come up with these methods. But the creativity I had from my art background really did come in handy. And I was given the time to look into the variables that impact test results that really has given us an insight into the industry today.
Yeah, I think that's one of the things that we all really love about the cannabis industry, is that it's such a frontier.
Yeah.
There's such, fertile ground for us to apply creativity and kind of innovative solutions to really push this thing forward. So that's really interesting. I had no idea you had an art background.
Yeah.
What specifically? You painter, sculptor?
Well, like I said, one of the minors is in art history. So looking back at all the different time periods and what drove art and what the artists were thinking at the times that they were creating the pieces that they created. But my studio art was centered first on drawing and then on oil painting.
Excellent.
Yeah.
We're gonna have to commission you to paint a mural for us in the office.
Well, my husband, it's funny. I recently came home with something that I did, and he told me that we couldn't have any more naked women in the house.
Oh no.
So apparently I'm not supposed to be doing the live modeling in the studios for a while, but I still do keep up with it, and I do really enjoy it.
Okay, excellent. Well, yeah, I'd have to check with HR to make sure we could bring that into the mural, but I'm not holding my breath.
Oh, well, I saw the picture of Kevin McKernan superimposed over the Grim Reaper's face, and I think that's I think that'll be just fine. That's some pretty heightened artistic ability right there.
There you go. Yeah, so wrapping up here, I know you said you were really excited to be coming to CannMed, and we're super excited to have you. If I remember correctly, this will be your first time out there, correct?
Yes, I've been chomping at the bit. CannMed is one of the most scientifically revered conferences that I've heard of. To me, CannMed and the Emerald are top notch. So it's an honor to be a part of the conference. I've heard nothing but great things about it. And I look forward to the scientific discussions and the collaborations that come about because of it.
Excellent. I think you're going to fit right in. There's plenty of people out there who kind of share your enthusiasm for cannabis science, and I think you'll be a great addition to the event. And we're looking forward to having you out there. And thanks again, Brianna, for taking the time to talk with us today. And if I don't see you before then, we'll see you out there in Pasadena.
Thanks so much, Ben. I'm looking forward to it myself.
I hope you enjoyed my conversation with Brianna Cassidy. Check out the links in the show description to learn more about the topics we discussed, and please also check out the CDX Analytics website. Our next episode will drop July 8th, but in the meantime, please go to CannMedEvents.com to view the Phase 1 schedule, see our growing list of sponsors and exhibitors, and sign up for email alerts. If social media is your thing, please also follow us on Facebook, Instagram, Twitter, and LinkedIn. Just search for CannMed Events. And lastly, if you are listening on a podcast app, go ahead and hit the subscribe button so that the new episodes automatically download to your device. And please also leave us a 5-star review. All right, that's it from us. Stay safe, stay healthy, and be sure to come back for the next episode of the CannMed Coffee Talk podcast.